So believe it or not, biochemistry is very redundant, and the organic chemistry mechanisms you'll see over and over again. And in this video, I'm going to go over most of them. So let's say we have this ester bond and this oxygen nucleophile. We know these can go through a chemical reaction where essentially we form a bond, and when we form a bond, we break a bond. So how do we do this? Well, the first step is we take this oxygen nucleophile and we deprotonate it. When we deprotonate it, we create a stronger nucleophile. This localized negative charge, so we have a stronger nucleophile. We also know this carbon is electrophilic. Why is this carbon electrophilic? Well, we know both of these oxygens are very electronegative. So therefore, they're pulling a lot of electron density. As they pull electron density, we create an electron deficiency on this carbon, so we know this carbon has a partial positive charge, so therefore it's electrophilic. So now we have a nucleophile and an electrophile, so they can react, where we nucleophilically attack this carbon. When we attack, we form a bond, and once we form a bond, we push these pi electrons up onto this oxygen. When we do that, we'd essentially form this tetrahedral intermediate, where again, we attacked, remember what we did. We formed a bond, and when we form that bond, we push these pi electrons up onto this oxygen, forming this tetrahedral intermediate with this oxygen anion. So now once we form this tetrahedral intermediate, we know this isn't stable with this formal charge of -1 that's not stable. So now what happens are these electrons essentially scoop down. They scoop down, forming a double bond. And once they do that, now this bond breaks where these electrons fall on this oxygen. So again, these electrons scoop down, forming that double bond. When we do that, this bond breaks where these electrons fall on this oxygen. So again, we form a double bond and we break this bond. So when we do that, we'd essentially form this product. So now we've done it. We formed a bond and we broke this bond. Then this isn't stable, and depending on the mechanism, it would get protonated at some point. But again, so we've done it. We formed a bond and we broke a bond. And this is a very common mechanism you'll see over and over again in biochemistry. And in fact, you'll see slight variations. For example, instead of this being an oxygen with this ester, maybe we'll have some kind of amide or amido. So again, it's the same idea, where again, we can take this oxygen and essentially form a bond. And once we form a bond, we break this bond with these electrons falling on this nitrogen. But again, it's the exact same mechanism, the exact same thing. The only difference is instead of an oxygen, this is a nitrogen. And again, when it gets protonated or not. But again, same idea. First step, we create a stronger nucleophile. Now we nucleophilically attack, forming a bond. We push these pi electrons up on this oxygen. When we do that, we form this tetrahedral intermediate. Now the electrons scoop back down, forming a double bond. And when that happens, now this bond breaks. These electrons fall on this nitrogen. When we do that, we again have our product, where again, we formed a bond and we broke a bond. And again, this would be negatively charged, but the point is that then it would get protonated. But again, we formed a bond and we broke a bond, and it's that exact same mechanism. And you'll see the same general mechanism over and over again in biochemistry. And again, we know this could be an oxygen, it could be a nitrogen, it could also be a sulfur. But again, it's the exact same mechanism. We form a bond, we break a bond. And again, we know how we do this now. Create a stronger nucleophile, we nucleophilically attack, forming that tetrahedral intermediate. Then electrons scoop back down, forming that double bond, breaking this bond, these electrons fall on the sulfur. When we do that, now we form this, and then the sulfur would get protonated. And again, we formed a bond and we broke a bond. So you'll see, now that you understand and seen this mechanism, you'll see this mechanism over and over again in biochemistry. But we can also change it based on what this nucleophile was. In all these examples, whether this was oxygen, nitrogen, or sulfur, we could have done all the same mechanisms, but instead of an oxygen, maybe we had a sulfur nucleophile. But again, it's the exact same thing. Imagine those exact same steps, but instead of an oxygen, now we have a sulfur. So again, first step, create a stronger nucleophile. Now we have a stronger sulfur nucleophile with that localized negative charge. Then it's the exact same thing. Nucleophilically attack, forming a bond, pushing these pi electrons up on this oxygen. When we do that, we form this tetrahedral intermediate. Then the same thing. Electrons scoop back down, forming a double bond. Now this bond breaks, these electrons fall on this oxygen. And when we do that, now we form this, and then again the oxygen will get protonated. But again, we formed a bond and we broke a bond, but it was that exact same organic chemistry mechanism. So it's really redundant. You'll actually see this over and over again in biochemistry. It's the same tricks. But now we saw it can be an oxygen nucleophile, it could be a sulfur nucleophile, but it could also be a nitrogen nucleophile. And if it's a nitrogen nucleophile, how does this mechanism work? Again, it's the exact same thing. First step, create a stronger nucleophile. Then we nucleophilically attack, forming a tetrahedral intermediate. Then the intermediate collapses. Then this guy gets protonated. But again, we formed that bond and we broke that bond. So the point is, you'll see the same general mechanism over and over again, but there can be lots of variation. For example, we know we can have variations in the nucleophiles. We can, for example, we had an oxygen nucleophile. We can also have a sulfur nucleophile, or we could also have a nitrogen nucleophile. So we had variations in this particular nucleophile, and we also had variations on this particular bond. Here we have an ester, but we can also have a thioester. And we can also have this amido type bond. But again and again, we can have combinations. For example, maybe we could have this amido type bond, and maybe we could have a sulfur nucleophile. But again, it goes through that exact same mechanism. We form a bond and we break a bond. And again, first step, create a stronger nucleophile. Then we attack. Then the tetrahedral intermediate collapses, where again, then this guy would get protonated. But again, it's that same idea. And it just varies based on the atoms, whether it's an oxygen, nitrogen, or sulfur. But again, it's that exact same mechanism. And you'll see these mechanisms over and over again. And what's important, essentially, is to realize that, again, there are these are three major types of bonds you'll see in biochemistry. You'll see these ester bonds, you'll see these amido bonds, and you'll see these thioester bonds over and over again, and you'll see these different nucleophiles, whether it's an oxygen nucleophile or nitrogen or sulfur nucleophile. And again, instead of this kind of organic alcohol, we could have also simply had a water molecule. But again, we have these different nucleophiles and these different bonds, and we can have lots of different combinations. This can break this bond, or maybe this guy could break this bond, or this guy could break this bond, etc. We can have lots of different combinations. So that's one common type of mechanism you'll see in biochemistry. But another very common mechanism is, again, breaking these phosphoester bonds, where again, it's the same idea. We form a bond and then we break a bond. So again, same idea. And the first step is, again, this is our nucleophile. So first step is we create a stronger nucleophile. So with this localized negative charge. So now we have a nucleophile. And also we know this phosphate is electrophilic. Well, how do we know this phosphate is electrophilic? Well, look at all these very electronegative oxygen atoms it's bonded to. All these oxygens are pulling electron density because they're more electronegative. So therefore we have a strong electron deficiency on this phosphorus. So now we have an electrophilic phosphorus. So now we have an electrophile and a nucleophile, so they can react. We nucleophilically attack. When we attack, we form a bond. And when we form this bond, we push these pi electrons up on this oxygen. When we do that, we form this intermediate, which is referred to as a trigonal bipyramidal intermediate. And again, the geometry isn't very precise here, but more precisely, the geometry would look like this, where again, we have this triangle, this trigonal, and then we have this bipyramidal. So this is more accurate, the geometry, what the geometry looks like, where again, we have these 60-degree angles, and then this is perpendicular. But again, the point is, we form this trigonal bipyramidal intermediate. Then once we form this intermediate, then these electrons scooch back down. When they fall back down, they form a double bond. And when that double bond is formed, now we break this bond with these electrons falling on this oxygen. When we do that, we'd essentially form this product, where again, we formed a bond and we broke a bond. So I know this reaction is a little complex. This mechanism is a little intimidating, but let's do it again. Remember, first step, we create a stronger nucleophile. Now the nucleophile nucleophilically attacks, forming a bond. We form a bond, and once we form that bond, we push these pi electrons up on this oxygen. So when we do that, we form, remember, this trigonal bipyramidal intermediate. We formed a bond, and then we push those pi electrons up on this oxygen, forming this trigonal bipyramidal intermediate. Now, once we form this intermediate, now we know the electrons scooch back down, forming a double bond, breaking this bond with these electrons falling on this oxygen. When we do that, now we'd essentially form this, where we broke this bond. So we formed a bond, and we broke a bond. And this is another very common mechanism you'll see in biochemistry. But again, just like we talked about earlier, we can have lots of different nucleophiles. We can have an oxygen nucleophile. We can also have a nitrogen nucleophile. Or instead of a nitrogen nucleophile, we could have a sulfur nucleophile. Again, we could whether it's sulfur or oxygen or nitrogen, it's the exact same mechanism. First step, we create a strong nucleophile. Now we nucleophilically attack, forming a bond, pushing these pi electrons up on this oxygen. So when we do that, remember we form this trigonal bipyramidal intermediate. Then the electrons fall back down, forming that double bond, breaking this bond. These electrons fall on this guy. So when we do that, we form our product. We form a bond, and we broke a bond. But again, it doesn't matter what kind of original nucleophile you had. Whether it was a sulfur nucleophile, or an oxygen nucleophile, or a nitrogen nucleophile, etc. It's that exact same mechanism. And you'll see this mechanism of a nucleophile forming a bond with one of these phosphoesters and then breaking a bond. You'll see that over and over again in biochemistry. Now that you know this, you'll recognize it. So for example, a very common macromolecule in biochemistry are these phospholipids, where we have this glycerol backbone and these fatty acids and this phosphate. But again, now with our mechanisms that we know how we break these bonds. We know how we break these bonds. For example, whether we have an oxygen nucleophile or a nitrogen nucleophile or sulfur nucleophile. And again, for simplicity, let's just create a very simple oxygen nucleophile as in water. And again, we have a lot of water in the cell, so commonly water is the nucleophile. But we know using this water, we can form a bond and we can break a bond. And we know we nucleophilically attack, pushing these pi electrons up on this oxygen, forming that tetrahedral intermediate. Then the electrons scooch back down, reforming that double bond, breaking this bond with these electrons falling on this oxygen. But again, we can essentially form a bond and break a bond. And that's how we hydrolyze those ester bonds, and now we formed our product, where again we formed a bond and we broke a bond. So this bond that was formed is represented by this bond, and then we broke this bond. So that's how we hydrolyze these ester bonds in these fats. But not just that, we know we can also hydrolyze this bond, where again we know this mechanism. We nucleophilically attack, forming a bond. When we do that, we push these pi electrons up on this oxygen, forming that trigonal bipyramidal intermediate. Then the electrons scooch back down, for reforming that double bond, breaking this bond with these electrons falling on this oxygen. Again, we're forming a bond and we're breaking a bond. And when we do that, we'd essentially form this product, where again, we broke this bond. That's how we hydrolyze these phosphoester bonds. And again, it's that same mechanism that we learned about earlier. And you'll see these kinds of mechanisms over and over again. So now let's do another example. Let's do another example. Let's say we have this dipeptide, where we have these two amino acids linked together with one of these peptide bonds. Again, remember, it's one of those bonds we saw, where again, whether this is oxygen, nitrogen, or sulfur, we know how we break this bond, as long as we have a nucleophile. And again, for simplicity, let's just use a simple water nucleophile. But we know this mechanism. We nucleophilically attack, forming that bond, then we push these pi electrons up on that oxygen, forming that tetrahedral intermediate. Then the electrons scooch back down, reforming that double bond. Now we break this bond, these electrons fall on that nitrogen. But again, it's that same idea. We form a bond and we break a bond, these electrons fall on the nitrogen. And now we know how we hydrolyze these peptide bonds. That's how we use water molecules to break these peptide bonds. And now we broke the peptide bond, where again we formed a bond, and that's represented by this bond. And then we broke this bond. So now we don't have this bond anymore. So that's how we break these peptide bonds. But again, it's that same mechanism that we learned about earlier. You'll see these same mechanisms over and over again. For example, maybe we have this nucleotide, where we have these two nucleotides, dinucleotide, linked together. We know we can essentially take one of these nucleophiles, whether it's an oxygen nucleophile, nitrogen, sulfur nucleophile. We know we nucleophilically attack, forming a bond. Once we do that, we push these pi electrons up in this oxygen, forming that trigonal bipyramidal intermediate. Then the electrons scooch back down, reforming that double bond. Now we break this bond. These electrons fall in this oxygen. But again, we know we form a bond, and then we break this bond. These electrons fall in this oxygen. And when we do that, we'd essentially form this product, where again, this bond that was formed is represented by this bond. And then we broke this bond, so that's why we don't have this bond anymore. But again, it's that same mechanism. Whether it's a phosphoester bond we're breaking or an ester bond, now we know. So again, in biochemistry, you'll see these same mechanisms, whether it's lipids or proteins or nucleic acids. With these bonds, now we know how we break these bonds. We break these bonds with these nucleophiles, and whether it's an oxygen nucleophile, sulfur, or nitrogen nucleophile, it's that same idea. Whether we use this oxygen to break this bond, or maybe we use this sulfur to break this bond, or maybe we use this sulfur to break this bond, or we use this nitrogen to break this bond, etc. We can have lots of different combinations. We can have different combinations of what breaks what, but again, it's that same general mechanism you'll see over and over again in biochemistry.